A little while, but here are our notes on cellular respiration. This will conclude Unit 3. So let's get started. So before we move on, all cells transfer the energy that is found in organic molecules, biomolecules, things like sugars, lipids, etc., to ATP, and they do this in a process called cellular respiration. All living things can perform cellular respiration anaerobically, meaning without oxygen. However, eukaryotes and a few prokaryotes can perform a more efficient version that is aerobic, using oxygen that's able to extract a lot more energy from each molecule of glucose than anaerobic respiration. So what is cell respiration? It's a set of enzyme catalyzed reactions. So there are a lot of enzymes involved. Don't worry, you're not going to need to be able to name any of them or memorize any of them, although you may see diagrams or descriptions of experiments that include some of these enzymes. And these capture the energy from biological macromolecules and store it ultimately in ATP. You end up getting a lot of energy released when you break molecular bonds between two carbons as well as carbon-hydrogen bonds. Now, this is a process that occurs in a lot of small steps, and the reason for that is if you look at this diagram, if you were to directly burn sugar to release energy, you'd have to put in a large input of energy, and then you would have a lot of energy released at once, which would be very difficult to harness and use for biological processes. On the other hand, if you do this in a bunch of smaller steps, one, you need less activation energy per step, and two, you also are able to extract the energy in smaller amounts, which is more controlled and allows you to store it. So let's talk about aerobic respiration. And remember, aerobic literally means air. In this case, we mean oxygen, and it uses oxygen for this process. It has three linked processes that take place. There's glycolysis, which is going to initially break down glucose and form two smaller molecules called pyruvate. That is going to form some ATP, as you see in this diagram right here. It also releases some high energy electrons, which are going to be carried by a different high energy electron carrier from what we saw in photosynthesis. In photosynthesis, we saw NADP plus and NADPH. In cell respiration, we're going to end up having NADH and FADH2. All right, and then what happens is you have something called a link reaction or pyruvate oxidation. You're going to take that pyruvate and you're going to convert it into something called acetyl-CoA. Acetyl-CoA is going to have two of those carbon atoms from the original glucose molecule, and then you're going to end up releasing one carbon atom in the form of CO2. And then finally, that acetyl-CoA is fed into what's called the citric acid cycle, also known as the Krebs cycle, and that's going to end up converting the two carbons that we had coming in that acetyl group into CO2. So it's going to release two CO2 molecules. It's also going to create some ATP as well as storing some energy in the form of those high energy electron carriers, NADH and FADH2. And then you also have those high energy electron carriers are going to be fed into the electron transport chain, which is a process we'll go over in a little bit called oxidative phosphorylation. This produces about 32 molecules of ATP for every molecule of glucose that you put into the system. So glycolysis is a process that happens in every cell on the planet. Because it happens in every cell on the planet, it can't happen in mitochondria because remember, not every cell on the planet has mitochondria. So this happens in cytoplasm, or technically in the cytosol of all cells. You don't need to know any of the intermediates. You don't need to know any of the molecular details of what's going on. What's really important is that you know what goes in, what comes out. So one thing to keep in mind also is that you do have to add some energy to get this process started. So you do input 2 ATPs and then you get 4 ATPs out. So you have a net gain of 2 ATP molecules. So in the end, you produce 2 pyruvates, 2 ATP when you subtract out the 2 ATP that you used, and 2 NADHs, which are storing some of those high energy electrons. So let's look at this process happening in an actual mitochondrion. So just a few things to note that we're going to start out in the cytoplasm. This hexagon that you see here, that's a glucose molecule. Notice we also start with an NAD+, which has no energy. None of this process for glycolysis will happen in the mitochondrion. It's just I'm going to be using the same diagram for all the other steps of cell respiration, some of which will happen in the mitochondrion. So first, we have that glucose molecule is going to be split through the process of glycolysis. It's going to make 2 3-carbon sugars called pyruvate. It's also going to make some ATP as well as those high-energy NADHs. The next step is called pyruvate oxidation. It's also called the link reaction because it's the link between glycolysis and the Krebs cycle, which are both processes that are going to produce some energy. Although you do have a little bit of energy produced here as well. So what happens is the pyruvate from glycolysis gets moved inside the mitochondrion. And once it's inside the mitochondrion, it's going to get converted to something called acetate, which is basically a 2-carbon group that's going to be attached to something called coenzyme A. Once it's attached to coenzyme A, it's going to form something called acetyl-CoA. Acetyl-CoA is then going to be fed into the Krebs cycle, which we'll talk about in a little bit. And you also will produce another NADH here with one of the high energy electrons from this process, from the pyruvate. And then you'll also release a CO2, which is part of the CO2 that's released by this process. So these are the end products. And now we're going to end up inside the mitochondrion. So going back to what we had in our previous diagram, we had our 3-carbon sugar, our pyruvate. It's going to move inside the mitochondrion, and then it's going to be joined with, through the process of pyruvate oxidation, it's going to be joined with the coenzyme A and form acetyl-CoA. You have 1 carbon removed, that's going to form carbon dioxide, and you have some energy stored in the form of NADH. All right. The next step of the cell respiration process is the Krebs cycle, otherwise known as the citric acid cycle. Just like the Calvin cycle, it's got a couple of names. You don't need to know any of the intermediate molecules that are involved. You don't need to know the enzymes that are involved for the AP test. What is important to note is the fact that you're taking in 2 carbons via that acetyl group. You've got those 2 carbons then being released in the form of CO2. You're going to make some ATP and you are going to make some NADHs as well as FADH2. So this happens in the matrix of the mitochondrion. And basically all the things that I talked about. The energy that is producing ATP is going to be stored in that ATP by something called substrate level phosphorylation, which, if you remember the worksheet that you guys did in class, was one of the questions. It was asking which of the parts of cellular respiration are going to form ATP by substrate level phosphorylation versus oxidative phosphorylation. And then in the last step, you're going to regenerate your original molecule that started out this cycle, so that way you can run this cycle over and over again. These are the products, and here let's look at it going on. So we've got our acetyl-CoA, which was sitting in our mitochondrial matrix. It's going to then be fed into the Krebs cycle, aka citric acid cycle. You've got carbon dioxide coming out. Here is your acetyl group going in. Here's your carbon dioxide coming out. And notice we're storing energy in the form of NADH. I also have here, you may notice GTP, but we're going to just replace that mentally with ATP because we're not going to get into those details. You also have FADH2 being produced. So we made a lot of NADH and a lot of FADH2. Well, what's the purpose of that? The purpose of that is these guys are what are called coenzymes. They carry high energy electrons. They pick them up, they drop them off, and in the process, they carry energy around. And this also includes, if you look at this group in general, it also includes NADP+, aka NADPH. And each of these molecules has two forms. One where they do have energy and they're carrying the high energy electrons, and one where they do not have energy and they are not carrying the high energy electrons. In general, what I think of is I think of the H in those molecules as standing for high energy. So that way I can remember which form has energy, which form doesn't. And by the way, you don't need to know anything about the structure of these molecules. I just included the pictures on the previous slide because I like to have lots of images. All right, so what's going to happen then is these high energy electrons are going to be dropped off to the electron transport chain. We've got another one of these, just like we did in photosynthesis. So you see here, NADH is dropping off its high energy electron, FADH2 drops off its high energy electrons, and then those electrons are going to have their energy used to actively pump hydrogen ions across the inner mitochondrial membrane. We're making another proton gradient. So we now build a concentration gradient of these hydrogen ions, and then when those hydrogen ions diffuse back through the membrane, they do so through that familiar molecule ATP synthase. And ATP synthase uses that movement of the hydrogen ions to create ATP. This is called oxidative phosphorylation. And then one thing to note is that these electrons that are coming out at the end, they have to go somewhere. They can't just be shoved into the inner mitochondrial matrix. They need an electron acceptor, and that electron acceptor ends up being oxygen, which we're going to talk about in a little bit. So a few notes about the proton gradient. We have that highly folded inner mitochondrial membrane to increase the surface area, so that way we have more molecules of the electron transport chain and more molecules of ATP synthase available to be able to maximize the amount of ATP production that we can have. In prokaryotes, since they don't have multiple membranes and they don't have any internal membrane-bound organelles, what ends up happening is those protons are going to flow across the plasma membrane. And just keep in mind that with that hydrogen ion concentration gradient that's formed, the concentration of hydrogen ions is going to affect your pH. So the pH inside the mitochondrial matrix is going to be higher than in the intermembrane space. So let's look at this process in action. So we have a few things in here. We have oxygen in the center. I'm not marking on this so it doesn't mess up with any of the animations. We have FADH2, which is carrying high energy electrons, and we have NADH, which is carrying high energy electrons. And these molecules that I highlighted are the electron transport chain, and this molecule is our ATP synthase. So we have our NADH come. It's going to drop off its high energy electrons. Those high energy electrons are used to pump hydrogen ions across into the intermembrane space. And then ATP synthase is going to allow the movement of those hydrogen ions, and that movement is going to produce ATP. You have the same thing happening with FADH2. Technically, they both go into the same electron transport chain, but I didn't want to confuse things, so I'm showing them going into separate ones. That energy is used to pump hydrogen ions across the membrane, and then you end up having the hydrogen ions go through ATP synthase to make ATP. And those electrons, those high energy electrons, end up getting combined with that oxygen to make water. So let's talk electron acceptors. When you are producing NAD+, which is going to be the low energy form, and you've removed those electrons, what you end up needing is a final electron acceptor, which is oxygen. And that oxygen, once it accepts the electrons, is then going to be converted into water. This is the chemical reaction where that happens. And so the key role of oxygen in these cells in aerobic respiration is to act as the final electron acceptor. This is not possible in anaerobic organisms that don't use oxygen. So what they'll use is they'll use some other molecule as a electron acceptor. So let's talk about that anaerobic respiration. Anaerobic respiration is also called fermentation. It's anaerobic, meaning it does not require oxygen. In fact, a lot of anaerobic organisms are poisoned by oxygen. It produces other molecules as the waste product or one of the final products of cell respiration, such as alcohol or lactic acid, depending on which type of fermentation it is. You end up converting your NADH back into NAD+, and basically what you're trying to do is regenerate that NAD+, so that way you can go ahead and then feed that NAD+ back into glycolysis to pick up some more high energy electrons. Without this final electron acceptor, your NAD+ would end up your NADH would end up staying as NADH. You'd never have more NAD+, and so you wouldn't be able to continue picking up high energy electrons. This does not give you any additional ATP generated, so it's very low yield of ATP per glucose molecule. So if we look at, for example, lactic acid fermentation. Lactic acid fermentation basically takes pyruvate and turns it into lactic acid. And in the process, that pyruvate accepts the electrons from the NADH, returns it into the form of NAD+, which can then be fed back into glycolysis to pick up more high energy electrons. It's all about regenerating this guy. The end product is lactic acid or lactate, and this happens in a bunch of different types of microorganisms. This also happens in our cells as well. So when your muscle cells can't get enough oxygen during intense exercise in order to perform aerobic cellular respiration, in a pinch, they can use lactic acid fermentation in order to regenerate that NAD+, so they can perform glycolysis again. It's not as efficient though, so that's why you don't always just perform fermentation. Alcohol fermentation ends up converting pyruvate to another molecule called acetaldehyde, and that ends up becoming the electron acceptor, and that produces ethanol, which is the alcohol that, yes, is found in like liquor products and stuff. That is going to be the final electron acceptor, and that's going to regenerate your NADP or NAD+, so that you can perform glycolysis again. And its final product is ethanol. This happens in some yeast as well as can happen in some plant cells under anaerobic conditions as well. All right, sorry that was a long one. Hopefully future lessons are not as long. And next up, Unit 4.